All right. All right. So let's continue where we started. Some people asked me yesterday for references. I'm gonna upload these things and also the slides online. I haven't done that, but these are the sort of references that you'll see. So there's these two papers. You see most of the math that I'll talk about. I have talked about and I will talk about in these two papers. The first sort of the basics, not the experimental side, but the basics of population genetics single locus. You can find it in this review. I think this is very well written. This is a paper that I will tell you something about some concept today that relates evolution to non-equilibrium statistical physics and non-equilibrium thermodynamics. And it's very well sort of described in the supplementary material of this paper. So the main text is pretty much just words, which is nice to read and easy to read. But supplementary material, you can see all the math, and it's pretty nice. Then if we have time, and that's the goal for now, so to go into data and basically apply everything we've been talking about to real biological data. And so I will start with this one, hopefully at the end of today, and if we have time, partly going to the second one. The order is wrong. But yeah, so this is basically talking about evolution in time-dependent environments, and this is a coevolutionary system of phenotypes. Again, I'll put them online, and you'll have access to this, but just that you know. All right. So we sort of ended here yesterday. We wrote how to characterize evolution of mean phenotype in a very sort of hand-waving and messy way on the blackboard. I'll try to sort of characterize now a general. So the goal is that you get a sense of how to write these things in a general sense, right? So you find the sort of way of the machinery to derive these equations. And I'll sort of try to characterize or discuss what these distributions mean, why we care about the phenotype distributions and change of that over time, and so how can we relate these things to data? So we ended at the mean phenotype. We can write a similar thing for the variance of phenotype, and some people today ask me, so why don't we just write the whole distribution of phenotype and how that changes? And the reason for that, so you can do that. You can write this thing for basically generating function and solve it for generating function of distribution. It's going to be more messy. And the reason I'm doing sort of moment by moment is I'm going to connect all these things to observables that you can do measurements in the data. And you can see them in the data. And the goal is to, based on these observables, to infer modes of evolution. So what's the shape of the fitness landscape that your population have evolved in? And as a result of that, you see a certain statistics of phenotype. So normally, we have access to average phenotype in a population, the variance. Sometimes, variance is more sensitive to the number of samples you get, so because it's noisier. And we'll discuss about this, how the noise grows as you go further and further in moments. So once you're at the third moment, it gets pretty messy. And so you don't really have much data in that. So let's try to see what's the minimal amount of information we need to get something sensible about the fitness landscape. So that's why we're building this that way. The other thing is that, so I'm putting in a very specific shape of fitness landscape, which is just quadratic. And that you may argue that may be very specific. That's true. But first of all, you know how to change the shape of fitness landscape. But you can also think about the problem in a different way. If your population is locally sort of located somewhere in a very broad fitness landscape, locally what you care about, think about Newton's method, what you care about is the first derivative and second derivative, right? And so if you're not sort of very much distributed all over, and if your landscape is not, very spiky and singular and these things that normally life is not, then this gives you a good, at least a good approximation to approach this problem. So you can think about it as that way, and then you can also try to do a sort of tailor expansion of your fitness landscape, and this will be handy, and this gives you the sort of main contributions you should care about when you look at phenotype distribution. So these were just the disclaimers why we are looking at this very specific toy model. So it'll sort of prove to be pretty useful and insensitive to details. So that's the thing that makes it attractive. All right. So I told you that there is this covariance matrix, at least in this case, the variance of the phenotype. I gave you an argument why it should be proportional to the variance of phenotype divided by the population size, because just the standard deviation of the mean. But let's try to derive these variance values both for gamma, which was the mean phenotype, and for the variance, just from the first principles. And first principles would be from the genotype description. So what we had last time was we had a covariance matrix for sort of frequencies of genotypes, if you remember, G alpha beta, which was X alpha 1 minus X alpha, if alpha is equal to beta, so the diagonal terms and the off-diagonal terms, which were the product. So this is known as a sort of inverse of that. It's a Fisher metric. So it's a Fisher covariance matrix, so to speak. And so if we were to write down what's a G gamma gamma, which is the corresponding variance for the mean phenotype, we had a version of how to go from one noise in one representation to another one, namely from the noise in the number of individuals into noise to frequency. So this is basically more or less the same thing. It's going from genotype description to a mean phenotype description. All right, so the only difference is that it's high dimensional, so we need to sum over all the alpha and betas, but basically it's the same thing. So you end up having the sum, which is then the partial derivatives of gamma with respect to X alpha, which is the frequency of the genotype type alpha, right? And then gamma X beta, G alpha beta. So we know what gamma is. Gamma is sum of the phenotype alpha times phenotype of type alpha times the frequency of type alpha, right? That gives you the mean phenotype of a population. So if I do the derivative with respect to X alpha, that becomes super simple. I get what? I get E alpha, E beta, G alpha beta. All right. Let's do this. All right, so I can now put what G alpha beta is in this equation. So we know if alpha and beta are equal to each other, it's X alpha 1 minus X alpha, so that would be equal to E alpha E beta times sort of delta alpha beta X alpha 1 minus X alpha. And if they're not equal, it's minus X alpha X beta. All right. So this might look a bit scary, but it's actually pretty simple. So if I take the first term here, just the linear in X alpha, what I get out of this is basically I need to put beta to be equal to alpha because of the delta function, and I get E alpha square X alpha. So that's the first term here. And then the other term that I get is the one that's sort of coupled to x alpha square. And we have two versions of that. The one that tells us alpha and beta should be equal, right? And the other one that alpha and beta should not be equal, right? So that's basically all the alpha beta combinations, right? So you basically get alpha beta, E alpha, E beta, X alpha, X beta, right? When you sum the two terms with X alpha square, right, they're either equal to each other or not, and that's what you get. And what is this? Can anyone have? Well, this whole thing is a variance, and that's cheating because you know the answer. But so this term, you can split it, right? So it's a two independent sum. It's just E alpha X alpha times E beta X beta X beta. The two sums multiply to each other, and each of them is equal to the mean, right? So this is basically gamma squared, right? And this is the average of E squared, right? So this is the expectation value of E squared in the population.
Sorry, I missed. Why did you drop 1 minus x alpha?
Why did I drop? I did not drop one. So I take the first term, x alpha times 1, and this is what is here, times delta alpha beta. And the second term is x alpha times x alpha for cases that alpha and beta are equal. And then I have x alpha times x beta for cases that alpha and beta are not equal, and that's what this one is. Right? So you basically get out of this the expectation of E square minus expectation of E square, which is your variance. So G gamma gamma simply just is equal to the variance, and you can get that from the first principles. So we have three other things to calculate in principle, right? All right, so G gamma gamma, we got that. It's equal to delta. So you can do the same algebra, and you can show that G gamma delta is equal to the third moment of the distribution, unless third central moment of the distribution. So this would be the off-diagonal term of your covariance matrix. And so this is approximately equal to 0 if you assume that the phenotype distribution is Gaussian. Well, it is 0 if you assume it's Gaussian, but if it's approximately Gaussian, it's approximately 0. Right. And then there's G delta delta, which is basically the fourth moment minus delta squared. And if it is Gaussian, this would be 3 times second moment minus delta squared, which is equal to delta, twice delta squared. So the fluctuation of variance is proportional to the variance, right? Fluctuation of the mean is proportional to square root of the variance, so a standard deviation. And that has deep consequences for what we're going to write down. All right. So is this clear so far? All right, good. We calculated last time what the change in the mean phenotype is due to mutation and selection, right? We can do the same thing for variance. I'm not going to write down all the details. I'm going to tell you what the mutation effect is, and we can try to guess what the selection effect is, and you can try to calculate it explicitly. So the mutation effect, the change in delta due to mutations, there's a component that comes from sort of, all right, So this is just a linear part of it, which is, and I tell you in a minute what E0 square is. But we discussed what E0 square is as a scaling factor last time, but it appears here as well. And then there's a term that comes from eta calculus. And the reason that it appears is that, so if you think about delta, delta is the average E square minus gamma square, right? We know how gamma changes in time because overall, and what's the fluctuation of gamma is proportional to delta over n. If you have a thing that has power 2 and it's a stochastic process, you make a second derivative with respect to that and put the fluctuation of it as a sort of deterministic term for changes of that thing's square, right? So this is a consequence of eta calculus. So if everything was deterministic, this wouldn't have appeared. It's a good thing that appears because things don't blow up, and you'll see in the solutions. All right. We can try to guess what selection is. So, yeah?
If this is the result of an eta calculus, and if we use like Startinovich calculus, then did you get different thing?
No, you don't get different thing. Yeah.
So it's just
Yeah, you don't get different thing for the change of the probability density function, right? So, because you need to do the corrections at the end, right? All right, so then you have the selection component, right? So because I mean we're going from a Langevin equation to a Fokker-Planck, right? So the proper way of doing it is to use actually the eta calculus for this purpose, right? Yeah.
Can you please explain the third line? I then missed it. The G delta.
G delta delta. So G delta delta would be the fluctuation of delta due to sampling, right? So how we calculated G gamma gamma was going from genotype fluctuation and projected to the mean phenotype fluctuation. You can do the same thing from genotype fluctuation to the variance fluctuation, and it would be proportional to the fourth moment minus variance square. M4 is the fourth central moment of your phenotypes. So, and if you're in a Gaussian regime, the fourth moment is 3 times variance square, and that's what this thing is. All right, so the way we were writing selection was to connect this variance factor, the G gamma gammas and G delta gammas and all these things, multiply that by corresponding derivatives of your fitness potential, right? So let's write down what the fitness potential is. We had minus C0 gamma minus E star square, in this case, and then minus C0 delta, right? So this was our potential. So if I want to write selection effect, ddt of delta because of selection, the little s here, then I need to write down a G delta delta and then derivative of f with respect to delta. In principle, you need to put in also derivative of, in principle, this is the full thing, right? So you need to also do this. But this is 0. We just established that. It's the third moment. And so we know what that is. We know what this is, so we can actually calculate it. So derivative of f with respect to delta is just C0 minus C0, and then the other one is just 2 delta square, so we get minus 2 0 delta square. All right.
Sorry, can you go again through where you got this equation for selection?
So we had that last time, right? So the thing that we wrote basically was let's say we have a fitness potential and we have a metric or inverse of a metric, which is a covariance matrix in this sense. So the amount that you're going to change on your sort of phenotype manifold, now in the manifold it's the mean phenotype and the variance of phenotype. That's the space you care about for some reason. And so the amount you're going to change, you're going to go around, along your sort of surface. And that's sort of tuned or modulated by this G, right? And then the sort of the gradient basically tells you how much you go up. And the gradient, that's the gradient of your fitness potential in each direction. So in direction of delta, you sort of couple that with the inverse of your metric along delta, which is G delta delta. And in the gamma is the inverse of that in G gamma delta, so which is a diagonal of gamma and delta. And so the fitness gradient with respect to gamma. That's the intuition behind it. You could go from the very sort of the basics, right? So the basics was, right? So we wrote down such a thing, that d dt of delta is d dt of the integral of E minus gamma square W of E dE, right? So this is your variance, this is a derivative, the d dt, this guy goes in, and you get E minus gamma square, and fitness of E minus mean fitness, right? So this would be the sort of direct calculation. Yeah?
So just to sort of get the more abstract picture behind the formula you wrote down, basically what this is saying is that this time derivative due to selection is the gradient of the fitness function.
Multiplied by sort of whatever covariance matrix on your manifold, right? So you have to correct it on the...
So speaking in this differential geometry language, you
Yeah.
have to pull the index.
Yeah, exactly. So that's the key here. So that's where the G's come. Yeah. All right. So let me erase this here. Okay, so we have now two equations. So there's an approximate equation written over there, right? And there's one here. So there are two Langevin equations that we need to combine them to get a joint the Fokker-Planck equation for the joint probability density of gamma and delta, right? So I call it Q, the density. So the second order derivatives, right? So that come from genetic drift or the stochastic parts, right? So the derivative with respect to gamma should basically get you delta over n in front of it, right? So you get second derivative with respect to gamma times delta. You can actually take delta out because delta doesn't
Can
yeah.
you remind me what Q is?
It's the probability density of gamma and delta, joint probability density of gamma and delta across ensembles of populations, right? So gamma and delta are mean and variance of phenotype within a population. And now you run many populations and then you sort of look at probability density across populations. All right. So it's like P of X. So probability density of frequencies that we had before. So this is the sort of the noise stem from the gamma side. Then you have a second derivative for delta, which is the fluctuation of delta. And as you can see, this looks a bit more complicated because actually there is it has a multiplicative noise that's proportional to delta. So that's why the delta comes in. And then you get all the, so this is the operator, right? Is there any second derivative with respect to sort of a cross term? Do we get a cross term here? Yeah, the coefficient is 0, right? Because G gamma delta is 0. So basically the idea is that you put the G delta delta in front of the second derivative of delta, G gamma gamma in front of the second derivative of gamma, right? So the cross term is 0. And then you get the term, so let's just start with mutations. You get the term for mutation in gamma, which is 2 mu gamma minus gamma 0. And then the term for mutation in delta, which is 4 mu delta minus E 0 square plus this delta over n from E2. And then there are the selection ones. Um, 2C0 delta gamma minus E star. And then what's that? 2C0 delta square. Yeah?
Sorry, what's gamma naught?
Right. So maybe let me just define these things here. So gamma naught is just the random state of your phenotype, right? If you only have mutation, you want to go to the only random state. So it was basically 1/2 sum of EI, and EIs were the quenched variables that were sort of defining your phenotype, and then E0 square is sum of EI squares. Yeah.
Can you, so I don't get the step from the time derivative to that complex expression. Where do the different pieces come from? For example, the second derivatives, does that come from intercalculus or from the differential geometry picture?
So it's just so we have a so let's say for mean phenotype you have a Langevin equation here, right? You can write down this similar Langevin equation for the variance, right? So let's just write it down. Maybe that's more useful. Do you see here? So there's a mutation term which is minus 4 mu delta minus E0 square minus delta over n, and then minus 2C0 delta square. That was the selection. So these are the deterministic parts. And then there is a square root of 2 delta square over n chi delta, right? So that's the, I took out the variance from the fluctuations, right? And so you can now think about these two as joint Langevin equations, right?
This one and this one.
Yeah, this and that. And then if you think about the covariance matrix, right? So if you have a general covariance matrix and DW, right? So the joint Langevin equation is, let's say I have a vector x, I get an A of x plus dt plus some B matrix dw. This is your Wiener sort of process, and the B is your covariance matrix in a sense, right? So, or square root of that. BB transposes covariance matrix, and in what we have right now, our B is square root of delta over n, and the square root of delta is 2 delta square over n, right? Because the cross terms are 0 because of G gamma deltas. And you go from this formulation, you can derive that Fokker-Planck equation.
So
Yeah.
generically, the cross terms will be 0 if we assume a Gaussian
Yeah.
distribution of
Phenotypes.
phenotypes.
Yeah. Yeah. So the first two terms are noises, right? Gamma in gamma and delta. The third term is the mutation in gamma, right? Is this guy. The first term on the second line is the mutation in delta. It's the thing I wrote up there in delta, under the line, on the blackboard above, right? That's the mutation in delta. And then the last two terms are the selection factors. So the first one is the selection in gamma, which is this guy. And then the very last one is the selection in delta, which is this. Are we good? Okay, so this is a general form. You can write it for anything you want. It can be as high dimensional as you wish, right? So we wrote down a very high dimensional version of that last time for genotype distribution in general case, right? So it's exactly the same thing. Even at this level, this two-dimensional Fokker-Planck equation is hard to solve. One thing that you can look at, and you can sort of let's for the moment imagine there is no selection, right? So ignore the selection terms. You can characterize a probability current in this system, right? So d dt of q if this is a current operator, you can write it like this. And then you can try to see if, I mean, in general case, in the stationary state, if when you set, so if you set the stationary, if you put this to a stationary state, so this would be equal to 0, right? That means your current should be constant, right?
So the operator is acting on the product JQ or just on J?
This. Right, divergence of J, right? JQ. So in the stationary state, you can try to convince yourself from the shape of this thing that we wrote that your probability current is not curl free. So in the space of Q, gamma and delta, in the stationary state, you still live in an out of equilibrium state, right? Because there's no detailed balance. So if you have curl in the system, your if your probability current is absolutely 0, that's where you have an equilibrium. When it's constant, you have a stationary state. And so if it's constant and non-zero, and because it's probability, you can only have curls. And in this case, you would have curls in the space of gamma and delta. And so even the most boring projection you can build, and you would hope that because you're doing so much coarse graining, you get rid of all the currents of genotype, the curls that you produce in the genotype space, you still maintain the curl in the system. Yes?
I'm not entirely convinced by that. I mean, I can imagine pathological geometries that where you do not end up producing curl, right?
We can calculate it.
Oh, you can, okay.
So you can write it down as an exercise, and we can discuss that later. Let's take, I mean, we know what exactly our operator is here. You can characterize what your J is, and you can calculate what the curl is. It's a 3-line calculation, and it's not equal to 0. We were not convinced by that either. It was a surprise, but it's not 0. It's proportional to mutations, mutation times n, and it's a small number, right? But it's non-zero, and that means you can't do equilibrium sort of analysis for that, right?
Can it be 0 in some places and not? Or what is like, I'm just kind of trying to grab my head around this.
No, it looks like this. This,
Oh, so it's like, okay.
yeah. If this is gamma and this is delta, it's really not 0. All right. But the other thing we can do is we can try to sort of understand how the stationary state like scaling laws in the stationary state. So think about your statistical physics doing RG and trying to understand the scaling laws in some quantities and try to, yeah.
What's the boundary condition for this for comparison?
So delta can't go smaller than 0. It's a variance, and that's about it. Yep. All right. So I've been talking about the sort of scales, right? So E0 square as one potential scale in the system, right? And trying to characterize dimensionless quantities, right? So if you were to guess, how does the mean so we defined our phenotypes as sum of EI sigma i's, right? So there's a genome of some size L phenotype that's encoded by L loci. So now you can increase L, and the question is, how does your mean phenotype and the variance of the phenotype scale with L? Anyone has a guess?
Square root of L?
So, okay, how does the expectation of mean phenotype scale with L? Let's put it that way. That was not correct. The first one was not correct, right? So because we're talking about ensembles of populations. So now you can calculate expectation value of the mean, and how does that scale with the length of the genome? You can have a guess what the expectation value of gamma is, right, from this. So if, let's say you put mutation rate to 0, or you put selection to 0, either one. If you put selection to 0, it's very easy, right? So the mean is equal to gamma 0. And gamma 0 scales with L, right? So it's proportional to L. So we know the expectation value of gamma goes like L. So what about the expectation value of delta? You can do the exact same argument. Is it?
L squared? E0 squared.
It's sum of EI squares. But sum goes from 1 to L. Now, why is it L squared? It's L, right? So you're adding up L numbers. So it should also scale with L. So it's an expectation value of delta. You add one other EI square and then you get another number, right? So you don't get L square numbers out of it. So that also goes like L. Now you can sort of shift your phenotype statistics by their mean and define, shifted values. So we want to characterize how the fluctuations of these numbers scale. That's why we're doing this. So let's define gamma hat as gamma minus average gamma, and delta hat as delta minus average delta. All right. So now this requires a slightly sort of deeper intuition. How does the gamma hat scale with L? How does the fluctuation of the mean scale with length of the genome? So the idea is basically, if you go to very large L's, your mean should become very precise, right? What does that mean? It means the fluctuation shouldn't go linearly for gamma. It should be certainly sublinear, right? And from law of large number, it's probably square root of L rather than L, right? Because it's the fluctuations of the numbers, right? So so this is the average of across loci, sum of bunch of loci. So it should go, the fluctuation of that number should go as square root of L. And this is slightly more complicated, but what we can do is we can sort of try to build up an intuition from what we have in our Fokker-Planck equation. So we're looking at fluctuations, so let's ignore, fluctuations come from the diffusion terms, right? So we can basically ignore all the deterministic parts. We are shifting everything. So let's ignore these guys, and let's look at these terms. So what we know is that just do the scaling analysis. Whatever gamma fluctuation scale, delta should be square of that, right? Because Q is dimensionless, right? What you have here is delta over gamma square should be dimensionless at the level of fluctuations. So if gamma goes like a square root of L, delta should go as L, the fluctuations, right?
Could you repeat that?
Huh?
Could you repeat that?
Look at this term. Okay, so we know probability density, on both sides have the same scale. Whatever, it's 1 over a scale of gamma, 1 over a scale of delta.
Okay.
Right? So the only thing that should be dimensionless here is delta over gamma square. If gamma fluctuation goes as square root of L, delta fluctuation should go as square root as L, right? I mean, here these two have the same dimension. So you can basically do the transformation at the level of the whole probability density, and that's kind of something that I don't know how many of you have seen it before, but it's the thing you do in a sense. If you want to take out fluctuations. So let me write this down, do a rescaling in my variables, and write the probability density for the shifted variables, and rescaled by the things that I think should be relevant for them. So there's a space here that I'm going to fill in, right? So if I now scale gamma by square root of L, so that the fluctuations basically will be equal to 1, will be a number, right? So it will be dimensionless. So I want to put in dimensionless numbers here. So this would be L to the power 1/2 gamma hat. And then I want to have a dimensionless fluctuation here as L to the power -1 delta hat and T. Well, it's a stationary state, so there's no T. So is this complete? It's missing something, right? I need to put a prefactor because I sort of scaled the arguments. So the prefactor in front is L to what power? Anyone has a guess? +3/2. Close. Right idea. So this is basically how your stationary state scales, right? Gamma scales like L to the power 1/2. Delta fluctuation scale like L. And if you put sort of relationship, does this go up here? And if you try to sort of do an arbitrary moment in your system, right? Look at the expectation value of, let's start with gamma hat. Gamma hat to the power n. From the scaling relation, right, what we get is basically this should be as L to the power n hat, n over 2, right? The mean goes like L, but everything else go L to the power, whatever power you put in, divided by 2. For delta, this is a bit different, right? We don't have the 1/2, we have basically going as L to the power n. Now, in normal statistical physics, what we want to do is that we look at how fluctuations compare to the power n, maybe the nth central moment divided by the mean to the power n, so this dimensionless quantity, grows or decays if you increase L, right? And that means whether your system lives in sort of within the center limit theorem or not, right? So if I do L hat to the power n divided by gamma hat average to the power n, so this would be equal to L over n, which will be L to the power minus n over 2, right? And now if you let L to go to infinity, right, then this goes to 0. So in the limit of a large genome, the mean will basically approach it. So the values will approach the mean, right? Everyone will be at the mean value. But for the fluctuations, for the variances, right, so for delta hat to the n divided by delta hat average to the power n, this will be L to the n divided by L to the n, which basically is equal to 1
Even if n is
for any n, right? And that's the thing that's called coherent fluctuations, right? So your variance fluctuations scale like the variance, and you would never get rid of it even if you go to the L to infinity. And that comes from the fact that, you know, the noise in the variance is equal to delta squared. Yeah?
What if we put selection?
Well, the selection just shifts your mean, right?
So it wouldn't affect the.
It wouldn't affect the it's a deterministic fact.
Okay.
Yeah.
So in this expression for gamma hat, if I put n equal to 1 on the left-hand side, I get 1. On the right-hand side, I get something that
If you put n equal to 1, you get 0, right? Gamma hat to the power 1
is, yeah, right.
is 0.
Okay, I have another technical question for this, for the scaling factor in front of the probability distribution. On the left-hand side, the arguments have dimensions L to the 1/2 and L to n L, right? So the distribution should
You may be right. You're absolutely right. Sorry. How do I get this down? Yep.
Then I have a conceptual question.
Yes.
So you said that this probability term has a curl, so we have non-equilibrium steady state. Where does energy enter the system, / where is it dissipated, /
All right.
does that make sense?
Okay. The short answer, it doesn't make sense. The long answer is that this is not a physical system, right? So it's a stochastic process. There's no conservation of energy. And that's basically what makes it different from, so we should not think about it in terms of where the energy is going in, right? So we are looking at purely a stochastic process in which the mean of the phenotype, the changes of the mean is proportional to the variance, but the things that are changing is from the outside, and we have no idea how to model it. Like, this is not within the scope of the model, right?
Right, but still a non-equilibrium steady state should mean that I create entropy or something.
You will create entropy, and we are going to get into that. But you will have second law of thermodynamics without having first law. It's possible. Yeah.
It might be a trivial question, but I don't understand why the average of gamma hat to the nth power is also like L to the n.
So gamma hat itself, right, scales like square root of L, right? Now, because we have the scaling relation in the probability density, so you can sort of try to get the expectation values of these gamma hats. And the way they scale, you always get out L to the power minus n over 2 gamma hat out of that. And so the expectation value should cancel that out because these quantities should be dimensionless, the way we wrote it on the right-hand side, right? So here, so if I take this guy and put it to the power n, so this is dimensionless, right? Gamma hat L to the power minus 1/2. So put this to the power n, it should also be dimensionless. So that means gamma hat to the power n should scale like L to the n over 2 to keep this dimensionless.
Oh, yeah, I understand the I don't understand the denominator.
Oh, the denominator. So the expectation value of, gamma. Other than that, that's 0. Yes. So fluctuation divided by the mean, right? All right. Okay. So we have coherent fluctuation here, and that's basically what we get. The other thing that that it implies, and you can try to show that, is that the delta fluctuations, regardless of where your gamma is, regardless of your system size, the delta fluctuations are very fast. So you can treat them, in passing, I mentioned this last time, you can treat them as fast variables in a system, and you can then take this equation and marginalize it and basically use expectation of delta in there, and so look at the corresponding Fokker-Planck equation for marginalized distribution of gamma. The distribution of delta, right, so the changes of delta does not depend on gamma. So there's no gamma on this board, and that's basically everything that goes in to the delta dynamics, right? So you can basically split these two equations and characterize two Fokker-Planck equations for marginal gamma and marginal delta. And because they're one-dimensional these quantities, right, in that case you cannot have a curl, can't have curls in one dimension. So the stationary distribution will be equal to the equilibrium distribution in each case. And we know how to write equilibrium distributions. We've been doing this. And not surprisingly, you get basically the sort of Boltzmann type Q of gamma, equilibrium of gamma, that connects to the neutral distribution of gamma that comes from the first two like mutations, and the sort of impact of fitness, so fitness potential. So you basically recover the exact same formalism again. You can do the same equation for delta, get the same thing, it's just f of delta is different than f of gamma.
Sorry, how do you do the procedure of marginalization if I don't have the probability?
So you treat delta basically as expectation value of delta. So that means that it will be a number, right? And so it comes out of stuff, right? So you just move them out. Yeah.
The scaling, it should be L minus 1 delta.
Yes. All right. So the other thing that that you get, so now in the marginal distribution, you can characterize the equilibration time in the setting, right? So equilibration time is just a function of the deterministic part, right? So how fast you approach your equilibrium or stationary state. And in the case that mutations are small, mutation rate is small compared to the strength of selection, the time scale of approaching the equilibrium goes as 1 over the strength of selection. So this is, you can estimate what exactly the time scale is, right? Maybe that's a good exercise. But what this implies is that if I have so what is C? C is the curvature of this fitness landscape. So the large C, the sharper your fitness landscape, and the stronger your strength of selection, right? So if your C is very strong, it means you approach the equilibrium much faster. And the approach goes as 1 over C. So this is a time scale that will sort of show up later. All right. So to show you how things work, so this is the distribution of gamma that we just wrote down. Now putting in all the details, right? So this is what I call Q0, which only has the effect of mutations and genetic drift. Theta, as we defined last time, is just mu n. Was this thing called heterozygosity that you could go and measure in the genome. So this basically is a Gaussian distribution multiplied by another Gaussian, right? So the whole thing is a Gaussian distribution. It's shifted one. If you add selection, you shift from, you can look at it, right? So that's the distribution of gamma in equilibrium. The empty circles are in neutrality. So when you don't have the selection component, so you're equilibrating again, around some value that was the gamma 0, right? You're slightly broader. Now you turn on selection. You make your distribution sharper because everyone now wants to get close to your fitness peak. So you want to get closer to E star which is I think around 70 in this case, and you get sort of a tighter distribution. So delta becomes smaller and gamma approaches the peak of the fitness landscape. And it does that by this Gaussian shift. So delta distribution is a bit more interesting. So it has features that are similar to chi-square distribution, and it's because it's the fluctuation of the variance. So the delta zeros, so this is the neutral distribution of delta, which is the empty circles again up there. So it has a long tail distribution. Yes?
So in that case, you also assume gamma now is constant.
Well, the delta dynamics doesn't depend on gamma, right?
Oh,
Everything on that board, there's no gamma in there. So, but yeah, so you ignore the curl component, right? So that's where it comes from. Right, so it has a long tail distribution, right? Sort of a power law shape in the tails. But then once you turn on selection, you're going to turn on basically, you're cutting the tail of distribution in an exponential way, right? Because this is your fitness potential. It's C times delta or minus C times delta. And what it implies is that you get rid of the highly diverse sort of population and make them much tighter. That's what we see, right? So these are the two different variances. The one we see here is a variance of gamma across populations, so how the means of different population differ from each other. And on the right-hand side is how the variance within each population changes. So it's an intra-population variance versus cross-population variance or divergence. All right, so these were two different quantities, and they will sort of build up we're sort of trying to build up a measure that we can use to characterize shape of fitness landscape from data. So I mentioned there are two variances. There's within population variance of phenotype, the average delta, right? And then cross-population variance of the mean, right? So how much the means differ. And you can sort of try, and you can also calculate what the phenotype divergence is across time, in time. So if you're sort of at some mean value at some time 0, then you wait for time tau, how much your mean changes, right? And it's the Olsztyn-Uhlenbeck process, and you can evaluate that exactly. So it's still a Gaussian distributed function. So in the limit of tau goes to infinity, right, so this becomes the variance of gamma. All right, so there are basically two variances here, phenotypic variances. And then there's a genotypic variance, which was this mu times n, which was the number of sort of amount of heterozygosity you have in the genome. And we're going to cook up a measure that combines these things and form a dimensionless quantity, which is that thing up there, that is very sensitive to what fitness landscape you're putting in, but is not sensitive to what kind of encoding you have of genotype to phenotype. All right, so that's the quantities, this omega term that depends on time. It's a ratio of the divergence of phenotype over time across average across population and average of within population variance. So cross-population variance of the mean divided by within population variances. So it turns out that if you have no selection, if you multiply this by the mu n of the genome, if you have no selection, you get this curve, the gray curve up there, the neutral evolution curve, that basically saturates to value 1. It's a dimensionless quantity. Both numerator and denominator have the same sort of scaling with your phenotype. So it doesn't depend on sort of whether you're measuring height or you're measuring gene expression. It's dimensionless. And it saturates to 1, so you actually have a good measure of where the neutral curve is. So the x-axis is a divergence time tau in units of 1 over mutation rate. And if you put it in units of 1 over mutation rate, so the sort of saturation time in neutrality would be at value 1. All right. Now, if you turn on selection, what happens is that the gamma approach or the mean approach to optimum is much faster than the amount you squeeze the population. So the response of the mean is stronger than the response of the variance. And as a result of that, your sort of divergence of the mean will become smaller much faster than the variance. And you end up having a curve that is lower than your neutral evolution, so the blue curve. And the stationary state, or sort of in the limit of tau goes to infinity, you get a stabilizing selection sort of asymptotics of this curve that again goes as 1 over c. So the stronger your selection, the lower your curve, and you can read off what your selection, what the shape of the fitness landscape is by looking at these curves. Yeah?
Sorry, could you give me like a little more intuition for what this omega means about how the population, what's going on?
So, all right, so what's going on is we had the two variances in the previous curve, right? Sorry. So what I'm trying to calculate here is this is the variance of the, so let's think about the tau goes to infinity for the moment, right? So you look at the variance of the mean, which is the variance of this curve, and then you look at the mean of the within population diversity. So you run evolution, I don't know, 1,000 times, you measure mean phenotype, and you measure the variance of that mean across all this population. That gives you this thing. And then within each population, you measure how much diversity you have within a population, and that gives you average delta that comes from the average of this curve. And the ratio of these two, and as you can see, if you turn on selection, the variance of the mean becomes smaller. And the mean of the delta doesn't become that much smaller because it was initially a very long tail distribution. It's not very sensitive to where the peak is, it's rather very sensitive to the tail, right? And so you're cutting the tail, but you're still not doing as well as you're doing here. So this happens much faster than this one, even with the same strength of selection. So now if you compare the two, so the reason we're doing the ratio is we want to build up a dimensionless quantity. So I want to give you a number, but I don't want you to tell you if this is a phenotype is a height or it's basically, I don't know, gene expression level. And I want you to be able to say, okay, this was a strong selection or not. And this is the way to do it because the ratio wouldn't depend on the phenotype scale anymore. Is that clear?
Yeah.
All right.
But I didn't understand the factor 2 theta.
So the factor 2 theta basically is a way, because we, all right, so I'm sort of not telling you what the solution is, right? But we can calculate what the solution is. And the reason we are putting 2 theta in is to get the saturation curve for neutrality to be equal to 1. The 2 theta, you can estimate from, it's observable, right? So you can go and sequence the genomes, and you can basically tell me what the mu times n is. You might have a hard time to tell me what mu is. You will have definitely very difficult time to tell me what n is, but you can tell me what the product is. So we have 3 observables you can measure from data, and then you can tell me what the selection is, right? Other question?
So the phenotype without selection, does that basically track the fitness landscape?
Well, I mean, without selection, there is no fitness landscape. Selection is the presence of fitness landscape, right? So you can look at, there is no selection in this term. Right. All right. So there are two features here, right? So there's a saturation time and there's a level. You might not be able to, you know, if you don't have the 2 theta, you would still be able to tell me what the value should be because you know what the saturation time is. So you can actually try to guess from these curves what 2 theta is to make these two consistent, right? That's a trick we use in data analysis, because we sometimes don't have a good estimate of that, and we try to sort of make the curves consistent by just squeezing or elongating an axis. The other thing that will turn out to be the case, and I'll show you a bit of a sort of simulation result, is that I'm sort of keeping saying that, okay, these are dimensionless quantities, they're insensitive to details, and let's say how insensitive they are. So our phenotypes, this is epsilon for some reason, so were defined like this, right? Epsilon I, sigma I. Sigmas were the spins, epsilons were, or EIs were the phenotype. The effect of loci specific effect on the phenotype, right? So contribution to phenotypes. And you can imagine, okay, epsilons can come from any distribution, right? Is my sort of measure of selection sensitive to that? Because mean and variance I can calculate, mutation rate I can calculate in some way. Getting this thing out, if it is sensitive to that, the measure is completely useless, because you need to have pretty elaborate experiments to infer the EIs. And the answer is because we have large enough genomes or loci, so large meaning 20 base pair in some cases. So we sort of did simulation. Okay, so 20 is here. The answer is, okay, so this is already infinite length, and you don't care about the details of EIs, rather statistics of EIs. So you can do any type of annealed averaging across loci contributions, and because the ratio is dimensionless, the averages will cancel out, the annealed sort of statistics will cancel out, and the ratio will become insensitive. So this is a simulation that we did, taking this omega, asymptotic value of omega. You can do it for the whole curve, it doesn't matter. For different sort of distribution of these epsilons. So there's, I think the longest tail distribution in this curve is exponential distribution, it's still fine. If you go to a power law distribution, it doesn't hold. So that's basically the limit. And then the other thing that we did not have here was a recombination. So basically we assume the genomes are sort of completely asexually reproducing, right? Now you can sort of turn on recombination a bit in your system, and you would still basically get, so this is the blue ones are, I think the blue line is the infinite recombination, so you scramble the genome every round of population, right? So you just randomly shuffle the loci between genomes, and then the green line is a zero recombination, or the highest and lowest, and then all the other points in between is in between. So you can see that it's pretty sort of insensitive to even recombination, right? You can do epistasis, and if your epistasis is not something dramatic, you're also insensitive. So it's a pretty reliable measure altogether. So far, we have sort of covered parts of this phase diagram of evolution. So we discussed the first day, right, the regime of low mutation rate. So that was basically two locus evolution, two allele evolution, single locus, right? And we had genotype fixation in this case, right? You would start from genotype frequency. You characterize the fixation probability of different genotypes. You can have, in the case of transcription factor binding site story, you can have the distribution of the phenotypes in that population and very well characterize it. I showed you a figure right now for the case of infinite recombination. You can basically do a projection, and it's a very simple calculation going from the allele model to infinite recombination model of phenotypes, and basically calculate everything. It's just convolution estimates, and you get a joint distribution of gamma and delta. They split very well, and you can characterize it. And the other limit that is basically the more interesting one would be if you have no recombination in your system, and that's the calculation we have been doing. And so you don't have this you can't characterize fixation of genotypes. What you can characterize is the distribution of phenotypes in this case. And so this is basically where we ended, right? So we know the stationary state distribution of gamma and delta, and we know the marginal distributions because it's a legitimate thing to do because of separation of time scales. So any questions so far? How comfortable are you with the sort of this notation and the description we had?
To take some practice.
All right. Any urgent sort of, so we're going to move to non-equilibrium systems right now. We're going to sort of, so far everything we discussed was a constant fitness landscape, which was this quadratic landscape. And we are sort of using the same formalism and trying to characterize a case where your population is driven by some time-dependent environment. So your fitness landscape is changing over time, and that's the green line here. And your population, in principle, follows this environment. So this is, in principle, a more realistic case, right? The issue is we don't know necessarily what the environment has been in the past, right? So we have some records of drastic changes of the environment throughout, species evolution, but that's not what this environment really mean. What we mean by this environment is that how the population perceives the environment, basically what the population cares about. So you can go back to what Stephanie was telling you about, right? So it's basically how do you encode the environment in your biological machinery to then respond biologically to your setting, right? And basically, you might be able to do experiments with mouse and cat and do retina experiments, but it's hard to do it across evolutionary timescales and get reasonable answers about, dinosaurs. So what we do here is to treat environments in a very sort of statistical way. We're not going to characterize what exactly happened, but we say, okay, if I know some statistics of environmental fluctuations, can we say something about what we should see right now in our phenotype distributions? Okay, so the picture becomes three-dimensional, right? So we have a phenotype or trait distribution. So there's now the simplest extension to what we had before was before we had the fixed fitness landscape in green, right? So let's say the simplest case would be now I move the peak of my fitness landscape and I move it in sort of a fluctuating way. So there's a fluctuating peak over time, and there's a sort of fluctuation rate would be proportional to some, is a number which is constant. Alternatively, you can say I'm sort of changing the curvature of the landscape. That has different consequences, and we're not going into that, but you can calculate that as well. All right. So what happens is that, okay, so at a given time, your population lives happily in this fitness landscape, and then at the next time point, the environment changes, and the population follows the fluctuation of the environment. Now, time scales play an important role. If the equilibration time of the population, which we sort of mentioned what it is, does anyone remember? 1/c, right? So if the equilibration time of your population is much shorter than the time scale of your environment, so the driving rate of the environment, let's call it v, so the time scale is 1/v. So if it's much shorter, that means that you're living in an adiabatic situation, right? At every point in time, you have enough time to equilibrate in your local environment, and then sort of, a million years later, something else happened, and then you move again, right? So this is basically what's known as macroevolutionary fluctuation. If these two are of the same time scale, same level of time scale, this is microevolutionary fluctuations. The results, well, you can solve both in principle. and you will get different results, right? So you get different descriptions of the system. So the microevolutionary fluctuations, you basically can absorb them in your phenotype evolution and add some noise into it. And the shape of, for example, the omega curve won't change in that sense. It's just the level would change. So if you look at the mean phenotype in this sense, we can write down the evolution of mean phenotype as before. The only thing that happens is that our selection field is now a time-dependent, has a time-dependent component. Then we can do a joint stochastic process. Let's say we learned that delta fluctuations are fast, right? So we use that approximation still, and so this is now average delta, and everything is sort of we're going to do a marginalized description. So we can write down an evolution for the optimum of this fitness landscape. So its simplest version that doesn't blow up, right, would be an Austin-Uhlenbeck process with a driving rate V, right, that your E star, which is the peak of the fitness, can fluctuate in a broad range, and the range is sort of fluctuation is around some sort of value epsilon. So these two form now a joint evolutionary process, right? The interesting part is that this guy is following this one, but not the other way around, right? So it's sort of you can write a Markovian description of this process, right? And I'm not going into sort of all the I'm not going to do the derivation here, right? But you can try to do it. It's all written in the second paper I mentioned. But so basically, it's two coupled Gaussian processes that one is informing the other one or driving the other one. All right. I think for time, maybe. Depending on how far we go, I might go into this next time, but let me give you sort of a sense of what the results look like. You can write down the sort of the joint distribution of the gamma and E star. It's a joint Gaussian process. And you can look at basically the probability currents in this sense, right? And so in equilibrium, for gamma, where E star is constant, there's no probability current, right? And that's what you can see. So if you look at the gamma E star plane, you have a gamma that stabilizes around your E star. It's not perfectly diagonal, and that's the interesting thing. And the reason that it's not perfectly diagonal is that there are always mutations. So if you get very close to the fitness peak, there's always mutation that keeps you away from the peak. So there's always sort of a little bit of a lag between a population that is perfectly fit in the stationary state and the peak. So that's called a mutational load of a population. And that's basically this tilt of the distribution. If you go in the macroevolutionary fitness seascape in this case, right, so it's not landscape anymore, it's a seascape, what you end up having is sort of it's a similar diagonal, but then you have a probability current or a curl with a curl that basically goes around your optimum of the E star and is following the E star movement. So this is adiabatic. At each point you stabilize around the right value, but next time the value changes and you sort of still have enough time to stabilize correctly. That's why you're perfectly diagonal. If you're looking at microevolutionary seascape, right, so when your equilibration time is of order of the fluctuation of the environment, you have no time to sort of stabilize yourself around the right value. You distribute yourself around some average epsilon because the environment just moves so quickly, and your current just goes around that, which has nothing to do with your environment. But it looks as if you're stabilizing around something. It might not be the best thing you can do. So let's see what happens to our, yeah?
Actually, it is a case like when our equalization is much harder than 1 over, is not 1 order of magnitude. So
So if it's much faster, it is microevolutionary when it's of order of 1 over v. If it's much faster than 1 over v, it changes within your generation, right? So you just average over stuff. So the thing that matters would be from one generation to another, because that's where the mutations come from. Now, there's a group that works on phenotypic adaptation that adds this phenotypic responses. So in that sense, right, so if you have a way of changing your state without mutating in your genome, which is this phenotypic fluctuation, epigenetic, blah, blah, right, then you compare this time scale with the shortest time scale in your system that you change your state, right? So in this sense, the way to change the state by is by mutation, and that happens, by mutation and selection, right? So that's the tau ec, and so the fluctuation should just compare to that. All right. Okay, so what happens in the microevolutionary case, right? So adiabatic situation is you can again calculate what this ratio is, divergence over diversity ratio, multiply it. This is mu n is theta. So neutral curve is as before, right? So there's no selection, and also there's no fluctuation of selection. Easy. The blue curve shows the case that you have a selection coefficient C, but there's no fluctuation. So that's basically what we plotted before. Now let's say I turn on fluctuation with the same selection coefficient. So what do you expect? Up to time point, up to divergence time of equilibration time point, there's no difference between population in a constant environment and a population in fluctuating environment. So the divergence should exactly look like what we had before. But then as you go beyond equilibration time, you're observing the changes of the fitness landscape, right? And so what you end up producing is basically this linear increase in the divergence with a slope that is proportional to the driving rate of your phenotype. And this is basically coming out of the response to the environmental fluctuation. So you have the sort of the extra divergence over diversity ratio, this tau omega added adaptive. So this is called adaptation. You're adapting to an environment. So the omega add, that basically the difference between where you are and what you expect to be in the station under stabilizing selection. So omega stab gives you the amount of adaptation you had, right? So is this clear? Good. The thing that I sort of want to note on the side is that you can argue that, okay, if I turn on this sort of fluctuating environment, I might screw up the delta, right? So, and the answer to that is that no, because delta fluctuations are very fast, and they don't see the fluctuation of the environment. And this is the curve basically that shows delta for different values of selection strength under different driving rate. And you basically don't see any impact of driving rate on the diversity of the population. So everything that's happening is at the level of divergence, not diversity. Okay. So I think this would be the last theoretical thing I say, and then we can then focus on data next time. So that would give us a good sense of doing things. So let's say conceptually, let's try to characterize the total amount of adaptation that we had. So some of you know about sort of, I think, Michael Desai will talk about things like rate of adaptation at the end or speed of adaptation. You can try to sort of build an analogy between what we have been seeing and sort of statistical physics or non-equilibrium thermodynamics. And there's quite a bit of connection between the two ways of describing the total amount of adaptations in the system. So we're going to talk about entropy production, and but we start from the sort of equilibrium sense, equilibrium case. So I've been sort of discussing the time-dependent observables. And to build a time-dependent observable, you need to basically look at trajectories of your population. Now, phenotypic trajectories, you can do it for genotypes. So you need sort of time traces to characterize non-equilibrium states. For the sake of comparison, let's say we have a trajectory of gammas, our phenotype means, and it's an equilibrium case. What we are trying to do is basically, let's say I start from some value of mean phenotype, and my population lives in this fitness landscape with a peak at some arbitrary value, and I can sort of evolve over time. How evolution happens is that I climb up the fitness landscape step by step every generation, and the amount that I go up the fitness landscape is proportional to the slope that I see, right? So the amount I go up, so this is the slope, the amount I go up is the green arrow. And the total amount that I sort of adapt or would be the sum of all these green arrows throughout my evolution, right? From gamma 0 to gamma n. And in the case of fitness landscape, right, when the fitness doesn't change, it's basically equal to your final fitness minus the fitness at the initial point, right? It does not depend on how you increase your fitness. It doesn't depend on trajectory. It's just a state-dependent quantity. And that's what we know from sort of equilibrium statistical physics. So we sort of call this quantity, the sum of all these upward arrows, as fitness flux. So it's just a rate of adaptation in a sense, the flux that you had of the fitness over time. And so when you sum things up, it's a cumulative fitness flux. So it's a gradient of fitness times the amount of change in gamma that you have, right? So that's a gain in fitness you have. Now, let's say we have a non-equilibrium system. You start up from gamma 0, and you climb up the fitness landscape by the green arrow, and all of a sudden the fitness landscape moves. So you're here in the phenotype space, but then you find yourself in a lower fitness state. So then you go up again the fitness landscape. You're up here, you drop, you go up again, right? And then by gamma n, right, you have sort of the amount of effort you have put in is again the sum of the upward arrows, right? So the amount of adaptation that you have had, the sum of the green arrows, but it's certainly not equal to the final fitness minus the fitness at the initial time point. And it depends on your trajectory, and it depends on the history of your population. So there's a difference between non-equilibrium and equilibrium states, right? And so, but again, this is a robust quantity that you can characterize as a fitness flux, and it would be sum of the upward arrows. So you can do some interesting math, and that's the paper I mentioned to you to look at this supplementary material. And what will turn out to be the case is that this quantity has some deeper meaning, and it relates to basically heat dissipation or entropy production in a system. So heat dissipation is a sort of questionable quantity because there's no heat, right? But you can characterize entropy of these trajectories. So what we had in entropy, in sort of state distribution was you can characterize the distribution of states, the queue of states that we were calling, and then you turn on selection, and somehow there's an exponential term that relates the distribution under selection to distribution in neutrality, right? That was always the thing we were building up in the single allele case, in the phenotype case. So that means that the states in the selection, right? So if you're a good state, you're exponentially more likely to happen compared to the bad state that you would sample randomly in neutrality. Now, you can build up the same sort of, so in a spirit, this is a similar thing. So if you look at the trajectory forward in time, now time matters because we're an out of equilibrium system, and you can think about the change of the fitness as your out of equilibrium protocol, right? So you're changing the field that is driving the system. Now, if you look at the forward trajectory of the system in this driven state, and compare that with the probability of the backward trajectory in the same state, with the same drive, right? the backward trajectories are exponentially less likely than the forward trajectories, right? Because you're just going against the stream. And you can characterize the Kullback-Leibler distance between the forward and back, the probability density of forward and backward. In equilibrium, this is 0 because you have detailed balance. So they're as likely as forward is as likely as the backward. But in non-equilibrium, this is a positive value. And you can show that the sort of the expectation is equal to the expectation of fitness flux. Right? So this fitness flux appears in the exponent of this probability density sort of distributions. In the case that we have been discussing, so fitness flux in stabilizing fitness seascape, something with curvature that moves over time, you can calculate what exactly this quantity is, and it's equal to all right, let's draw this. So it's equal to the ratio of this adaptive value over stabilizing value. So if we have this curve, right, this was omega add, and this was omega step. This was going as 1 over c, and this was v times tau, right? So the slope was v. So if you calculate the fitness flux, it would be equal to the ratio of the omega add divided by the total omega, or omega step. Divided by omega step. So basically tells you how much adaptation you have compared to the sort of the range that your phenotype is distributed. So you remember, the less effective your selection is, the sort of shallower your fitness landscape, the higher your omega step would be, right? You go down with the selection. So if this is very high, then basically your adaptation is insignificant. You're fluctuating within the variance of your population. But if this is very small, it means that you're following the fitness landscape very closely. At each point, you're very much constrained, and every time the environment moves, you run after the peak of the fitness landscape. So the ratio of these two gives you fitness flux, and it's equal to V. So the cumulative fitness flux over time would be V times C times tau.
So it almost doesn't put it forward.
Say it again?
So V times C is like much less than 1, right?
Can you speak louder?
Yeah. V times C is much less than 1.
It doesn't have to be less than 1.
I'm treating the our equilibration is much less than. At some point we were in the regime, but like we were in when we were in adiabatic regime, right?
Right.
And then 1 over C was much smaller than.
So tau ec was much smaller than 1 over v, right? So this 1 over c would be much smaller than 1 over v. So it's not cv, but this c is much smaller than v, right? Yeah, so this value can be pretty large, right?
C is much larger than D, right?
Yeah, c is much larger. Sorry if I spelled it the other way.
What the axes were on the
So, right. So this was time. This was this omega, divergence diversity ratio, right? So this was this curve in a twisted form, right? So it's basically the ratio of the green divided by the blue, right?
So you are out of time.
Yeah, so I think we stop here, and next time we're going to look at data and use everything we learned to see what we see in data. All right, thanks.